If you learn one thing in a biochemistry class, it's that the structure of biomolecules, or the structure of any molecule, is going to influence its function. And this is especially true in proteins, where we have these long chain molecules that are made up of amino acids. And there's 20 different amino acids, but we can put them together in a dizzying array of configurations, and those different configurations and sequences give us all of the proteins that do all of the cool things in our bodies. So understanding the different levels of structure will help us to understand their functions and kind of how they work and why they work the way that they do. So starting at the ground level, the primary level, we just have the list of the amino acids that make up the protein. So if I just know the sequence, the order that they're in, sometimes this is described as beads on a string, so it kind of looks like a pearl necklace, for example, right? So we have kind of the different amino acids here. I have them represented with shapes. We could think about them in terms of their 3-letter designation, or we could think about them in terms of their 1-letter designation, and we just have this list that are bonded together, and we're showing these peptide bonds or amide bonds just with these linkages here. So I have a protein that's made up of this list and sequence of amino acids is called the primary structure of a protein. Now that's not really what proteins look like though, right? It's not just a sequence of amino acids, but it gives us a place to start. And there's some interesting features about them. So I'm going to give you a model here to look at because I think it helps to see this thing in 3 dimensions. So if this is my N-terminal end over here, this guy, so here's my N-terminal, and I have my amino acid kind of coming out, and then this is my peptide bond. So here's my carbonyl from the carboxylic end of this amino acid, and then we had a dehydration reaction that got rid of the water. Now we have an amine that linked up here, so that amine that's linked to the carbonyl gives me an amide linkage, which in the context of proteins we call a peptide bond. So that's what we're looking at there. If you look at this then, you can kind of see that my R groups are sticking off into space, but I have this kind of regular zigzaggy backbone that is made up of my nitrogens and carbons, and these double bonded oxygens from our carbonyl group are going to be sticking off in opposite directions kind of as I go, but my zigzag backbone here of my carbons and nitrogens make up the fundamental backbone of my protein structure. And this is rigid. This is a pretty rigid structure because the electrons in the double bonds of the carbonyl are actually delocalized and distributed with the bond between the carbon and the nitrogen. So this thing here kind of acts like some of those delocalized resonance kind of structures that you saw in organic chemistry, and this allows for it doesn't allow for free rotation. Usually we would say, well, this is a single bond, so I could rotate freely around it, but because of these delocalized electrons, that keeps this rigid. That means that all of this backbone are all in the same plane. So this part and my oxygens, these are all in the same plane of atoms. If I was to take a piece of paper and slice it through this whole thing, then I would have all of these atoms on that piece of paper, except for the R groups and some of these hydrogens that are sticking out in opposite directions. But that planar backbone of my protein is a good way to understand that primary level of structure. Now, just from a mathematical point of view, if there are n number, I'm looking at my writing, kind of looks like a frowny face the way that I've drawn it there. If there is n number of amino acids in a chain, so let's say that I have 6 amino acids. And that's what I have in this chain up here, but this doesn't really work as an example because I have alanine twice. But if I have 6 different amino acids in a chain, then there are 6 factorial ways that these 6 can combine together in that chain. So in terms of order, then 6 factorial, if you recall from math, is 6 times 5 times 4 times 3 times 2 times 1, which means that if I have 6 random amino acids and they're all different from each other, there are 720 ways that I could put those things together in various primary sequences. And just one of those is going to give you the right sequence to give you the protein that you're looking for. So again, there's kind of this dizzying number of combinations that we can put together for these various levels of structure. And this is just kind of the beginning. So here's a whole bunch of amino acids. We put them in a particular sequence. That sequence is the primary structure. If we build up, then we have that planar backbone of our structure. But now we have, from those planar structures, we have the carbonyl group from one amino acid, and we have the kind of amine group with that hydrogen that we said is sticking out outside of that kind of zigzaggy backbone. So this allows for the potential for hydrogen bonding. If we think about the dipoles on these things, there's a dipole here because the oxygen is more electronegative. There's a dipole here because the nitrogen is more electronegative. So that gives me a slight negative charge on my oxygen, a slight positive charge on the hydrogen from my amine side. So the hydrogen from my amine side and the carbonyl group from my carboxylic side from the various backbone amino acids have the potential to do hydrogen bonding. Recall that hydrogen bonding is an intermolecular force, meaning a force between molecules or between parts of a molecule in this case, where we have a slight positive and a slight negative charge due to the polarity of the bonds in there. And it's specific to hydrogen, hence the name. So hydrogen in a polar bond with either nitrogen, oxygen, or fluorine, and then the oxygen, nitrogen, or fluorine on a second molecule, or a second part of the molecule. So due to this hydrogen bonding, there are various secondary structures that can occur. So we had that sequence of amino acids. Now it's going to start to rearrange and organize itself and be attracted to itself, and you get a couple different features. One of them is called an alpha helix, and it kind of looks like a slinky, or I think about it like curling ribbon. So we have this structure in the amino acids. If this is my amino acid sequence, my kind of necklace of amino acids, then there's going to be some attraction between these things that causes this to coil up. So it coils up, and this is called an alpha helix. It's a Greek alpha. So that alpha helix always twists in the same way. So it's kind of a clockwise twist, and this happens because of the hydrogen bonding between these backbone structures. Another thing that can happen with this hydrogen bonding is it can cause these things to sort of fold upward. I think about this when it's really warm outside and you have a piece of paper and you fold it back and forth and back and forth, and then you can make a fan, right? That's kind of what this looks like. These are pleats. So we have some hydrogen bonding potential kind of between sheets here, between our different amino acids, and this attraction to these various things allows for these structures to occur. So we get this folding motion, and these are called beta pleated sheets. So pleated referring to the folds. That's a Greek beta there. All right, so these are my secondary structures. And again, this is occurring because of the hydrogen bonding in the amino acid backbone. But that doesn't take into account the R groups on my amino acids. So the R groups can account for all sorts of wackiness when it comes to tertiary structure. So that was secondary structures, which are specifically these beta pleated sheets and alpha helices. But when we get to tertiary structure, and I've kind of pre-drawn my molecule just because it's a little bit, it gets wacky pretty quickly, then this is the bending and folding of polypeptides that are due to these intermolecular forces, primarily between the R groups, but we also can see it because of the hydrogen bonding from backbone molecules. So if we have my N terminal down here, and I maybe have some beta pleated sheets here, this is a secondary structure within the tertiary structure, because the tertiary structure is all these bends and folds that can occur. Then if I kind of follow along here, I can see, well, in this section here, these are my backbone amino acids. So these guys can have some hydrogen bonding. And that allows for some attraction between different parts of the strand. And then within this pocket, there's all this hydrophobic action. So we have all of these just carbon to carbon bonds. So this is all hydrophobic. And we know that proteins are in a watery environment, biological organisms, so these hydrophobic interactions are going to cause this to kind of be attracted to itself, come in on itself, kind of like micelles if you've talked about detergents before. So these things like to be with each other, sort of a like dissolves like kind of feature, and they don't like water. So they're going to be interacting with each other here in the middle. So, you know, you have something like valine, or you have others that are just these hydrocarbons in the R group. They're going to want to interact with each other. Now we have kind of this interaction between an R group on one and the backbone on the other. So we can also have hydrogen bonding that way. So not just between the backbones, but between R groups and the backbones. We can have attraction between a negative charge on one and a positive charge on the other. This is called a salt bridge, referring to the fact that it's an ionic reaction or an ionic relationship. So the negative charge of one and the positive charge of the other are attracted to each other in a salt bridge. We have some more hydrophobic kind of interactions here. These guys like to be together, hydrophobic. So non-polar R group. And then we have what are called disulfide bridges here. This is a thiol group on either side. These come from cysteines, and these sulfurs can bond together. So we call this a disulfide bridge or a disulfide bond. So again, that's cysteine related, specifically because of that interesting sulfur characteristic, because these sulfurs like to bond together. So anytime cysteines look at each other, then they want to bond together and form these disulfide bridges. If we look up here, we can see again, so we had hydrogen bonding between the backbone, we had hydrogen bonding between an R group and the backbone. We can have hydrogen bonding between the R groups as well. So hydrogen bonding is kind of the name of the game for these things. And are we trying to get away from water? And are we attracted to each other? And are there sulfur interactions? And then we have kind of our alpha helix here, which is again a secondary structure. And then here's my C terminal with my sloppy oxygens. So this is tertiary structure. So the bending and folding of this chain due to all these intermolecular forces. And for some proteins, this structure, they probably don't look like this exactly, but this structure is the final functional form of the protein. So I could take this and I could use it in the body in this particular shape, which looks a little Dr. Seussy. This particular shape could be used for something, okay? And it's because of this interaction and because of the sequence and because of these particular bends and folds and secondary structures that allow for it to do what it's supposed to do. Now, if I need more than one of them, so that could be where we stop in terms of levels of structure. But if I need more than one of them, then I can go to quaternary structure, which is where I have multiple polypeptides that I put them together, and that gives a functional protein. The one example that people always use is hemoglobin. And if you see a picture of hemoglobin, it's a bunch of alpha helices. So here's one, there's one, and then you have another protein unit that looks like this. Here's some alpha helices and some bending. There we go. And then we might have another one down here, and some alpha helices and some bending and folding. And then we have another one over here, and some bending and folding. Not a biochemist by trade, I think, is the take-home message here, nor an artist. So now I have 4 functional polypeptide units that I've put in together, and then each of these has a heme group in them. So there's kind of an iron catcher's mitt in each one of these guys that can hold on to oxygen molecules. And then those oxygen molecules can be transported all over the body to wherever the oxygen is needed. So they act like little catcher's mitts in each of these polypeptides. And then that entire structure, that entire multiple protein strands act as the fully functional protein. But each one of those individually is not going to be functional. It takes all of those strands together that is going to cause that functionality. So that's quaternary structure. When I need more than one, then that's going to be the way that I put them together. Okay, so that's kind of a lot of information talking about these different levels of structure in proteins, but it's really important to understand kind of why they do what they do and how we get all that variability from them. So if you have any questions on this, don't hesitate to reach out. Otherwise, I'll talk to you again soon.